Okay, so the final speaker of this session is David Baker, and he's from the University of Washington, where he's a Howard Hughes medical investigator. And the title of his talk is Design of Protein Structures, Functions, and Assemblies.
All right, well, I want to thank the organizers for inviting me to this, what's really a fabulous symposium. This is the best best beginning to a symposium that I've been to in a long time. And again, congratulate Greg Winter for his achievements. Let's see. No, wait, what's going on here? Sorry, wrong talk. That would not be good. All right, that's better. All right, so I want to give you an overview today of the work we're doing on designing new protein structures, functions, and assemblies. And in contrast to what you've heard about so far, rather than obtaining new molecules by selection, we're doing it by computational design. And I need to emphasize, I put this slide on because sometimes after I give a talk, people come up to me and ask me if we ever do any experiments. So everything that I'm describing has this basic pattern. We have a problem we want to solve. We carry out a computer calculation designing a new protein with the desired structure or function. Since we designed the protein, we know what the amino acid sequence is. And so we have the amino acid sequence of this protein we've designed on the computer. And of course, with modern DNA synthesis technology, it's trivial to make many, many genes encoding the amino acid sequences of many, many designed proteins. It's a good thing we can make many genes because most of our designs don't work. There's still a lot. This is an early field, and we're continually able to improve our computational models using feedback by analyzing comparing the designs which work to those that don't. Anyway, once we have the genes, we make the protein in assay. So everything I'm describing here starts with a computation, ends with an experiment. The way the calculations work is shown in this animation for a very simple calculation that is designing a sequence to fold up to a given protein structure, in this case, the backbone shown here. So what's happening at each step in this animation is we're substituting in a randomly selected amino acid, a randomly selected conformation of a randomly selected amino acid at the position, evaluating the energy, and then accepting the substitution if the energy decreases. And in this way, we end up with sequences that are very low in energy in a particular backbone conformation. It's actually a little more complicated than that because for any fixed backbone conformation, there likely won't be a sequence which folds up to precisely pack in that structure. So we have to vary the backbone at the same time we're optimizing the sequence. So this gives us a sequence which is very, very low in energy for this backbone conformation, or the ensemble of backbone conformations which are closely related to it. But there's no guarantee that for that sequence, there isn't some other low energy structure. The way we address that problem is to take advantage of work that has been going on in my group for a long time on the ab initio structure prediction problem. And this is the classic problem from an amino acid sequence to protein three-dimensional structure. We've been working on this for a long time, but it's still a largely unsolved problem in that only for smaller proteins can we reliably predict the structure from the sequence alone. However, the proteins that we're designing are smaller proteins, and so we can make use of the methodology we've developed for large-scale sampling of the different conformations available to a polypeptide chain. We basically take the sequence of the protein, and actually what's interesting about the way we're doing these calculations is we actually send it out to volunteers all around the world who are part of the Rosetta at Home distributed computing project. They send us back the results for the lowest energy states they find for that sequence. And then we can compare the low energy structures that are found for that sequence in this large scale search of conformational space with the design structure that we're trying to make. And the results look something like this. These data were obtained by the 300,000 volunteers at Rosetta at Home. Each red point is the result from a single person's computer. What we've done here is to design sequences using sort of a somewhat more elaborate version of the design protocol I described that were particularly low in energy for one of these 5 simple globular protein structures. We took those sequences then and sent them out to Rosetta at home volunteers. And on this axis is the energy. On this axis is the distance from this conformation. And what these sequences have in common, well, first of all, what you see is that different people's computers found very different solutions for the lowest energy structure for this sequence. Some people predicted that the lowest energy was quite high and that the structure was quite far away from the design structure. But you see, with this large amount of sampling, we can get a pretty good map of the energy landscape for this sequence. And what you see is that the lowest energy sequences are very, very close to the design target conformation. And that's true in all 5 of these cases. Of course, for other sequences, they are not the lowest energy. We find other low energy states. And of course, those sequences we don't make genes for and we don't experimentally test. So this is proven to be a very stringent, you might think of an internal consistency check that we do carry out before any of the design projects I'll tell you about, that we require that the sequence actually not only be low in energy for the structure, but that when you do a search of all possible structures, or a very large number of structures, the lowest energy structure you find is this. So I think in part because we're being so stringent about how we select sequences, when we actually make the protein and solve the NMR structure, this was done in Guy Montaglione's lab, we find that these structures are very, very close to the design structures. These sequences are not similar to any sequence in nature. They're completely de novo designed. If you look on the right side, it's a little hard to see, but all the side chains are in exactly the right place. So we can design new globular structures with really very high level precision. And this has gotten to the point where a rotation student coming into my group for a couple months can basically go through the design, pick a new type of structure, go through the design calculation, make a gene, and show the protein stable in the time course the rotation. The structural biology usually takes a little bit longer. So this works quite well for a quite wide range of monomeric protein domains. We've done this for domains which have sort of where there are topologies like that seen in nature, topologies where there aren't seen in nature, and but those are single domains. And how do we get more complicated structures? Well, we can start fusing together these elemental building blocks to get larger structures. And this is just an example of this. This is a model of fusing these two de novo domains together and redesigning the interface. And this is a crystal structure, and they're basically identical. So we can work our way up from scratch, starting with sort of these simple domains, and then make more complicated things where you can start thinking about designing active sites and so forth by putting them together. I wanted to mention the work of Vikram Mulligan, who is a graduate student here, who's now extending these ideas to design cyclic peptides, very, very small peptides. And he, in this case, he can do full multi-state design and find explicitly designed for sequences that are stable in the desired state and not stable in any other state. And this just shows early experimental results from designs he's characterized where the NMR structures that he's solving are very, very close to the design models. And he's going on now to do this with mixed D and L amino acid peptides, with bicyclic peptides and other types of things. And these are obviously structures where it's a little bit harder to do selections, and so I think there's a lot of promise here. Now, as I'll come back to later, once we had Rosetta at home, I realized that there's a lot of people out there who are smart and can help you out in your research. And so we started a project called Foldit, where we enlist volunteers from all around the world to design new proteins. And I really like these. We gave people an extended chain of 70 amino acids and told them to fold it up and design and change the sequence. It was just a polyalanine 70 residue chain to fold it up, design the sequence, and make something that would fold up to a trimer. And we set it up so it's symmetric. So they just had to design one monomer, and then the whole thing assembled on the computer, the trimer. And the graduate student who's been working on this, going back and forth with Foldit players, has just gotten up his courage to order genes encoding these designs. So I can't tell you whether they work or not, but I think they're quite beautiful. So I think we have the general public doing protein art. And you can see some of these really don't look like anything that you see in nature. I'm quite excited to see this. And more generally, if you want to design proteins, all you have to do is sign on to Foldit. Okay, so that's where we are on designing new protein structures. And I should just say there are other types of structures, like I've been working on very long helical bundles and trying to make like cylinders, and we have some very exciting results on those, as well as repeat proteins, or de novo designed versions of the kinds of things that Andreas talked about. But I don't have time to talk about that today. Instead, I wanted to go on and talk about, in the next part of my talk about design of function. And I'll start by talking about design of protein-protein interactions. And here the idea is if you have a target structure shown in the surface view, we really start with a concept that was pioneered by Jim Wells, who's here, which is to try and identify interaction hotspots, ways in which side chains could interact, disembodied side chains could interact very strongly with this surface, so fit into nooks and crannies. And so that you might think of as, if this was a climbing wall, the surface here, those side chains would be like the climber's hand and feet fitting into handholds and footholds. And then the next step is to fit a body on here, a protein scaffold, which can hold all of these side chains in place. And just to illustrate it, this is work that we published a couple of years ago. This is just a blow-up of a site from the influenza hemagglutinin, a conserved site in the stem region. And this just shows side chains fitting into a conserved groove in the structure, which is in yellow. And then in pink are different small helical protein scaffolds that hold these side chains in the right configuration. When we make these proteins, they bind the virus, and Ian Wilson's lab solved structures of these bound to the virus, and they bind exactly where they're supposed to bind. This is the one that was on the left, the one that was on the right in the preceding picture. When you look at the interface side chains, they line up almost exactly, the design. So the design's binding in exactly the way the computer model said. So we've gone on since then. Eva Strauch has gone on to design binders to the head region, to the sialic acid binding site. She's used a similar trick to what Andreas mentioned. She's actually put a trimer, designed a trimer that holds these in exactly the right place to interact with the 3 sialic acid binding sites on the flu. She calls it flu glue because, as in Andreas's case, this protein does not seem to come off the flu virus. So now we have a series of binders down here and a series of binders up here. And one of the immediate applications is for diagnostics, because you can capture with one and read out with the other, for example, in a kind of a paper strip format assay, which we're collaborating with people at the University of Washington on. So with different binders to these different sites, we can actually cover a pretty wide range of existing flu strains, and this is increasing by the month. We have very preliminary results with collaborators at the UW, spraying these proteins into mouse lungs and then challenging them with flu virus. And the initial results are very encouraging, with flu-infected mice losing body weight fast. But if they're pre-treated with these binding proteins, that doesn't happen. Much more work needs, of course, to be done along these lines. So we don't have to target just pathogen proteins. We can also target self-proteins. So this is the PD-1 protein, and we've designed a small protein which binds quite tightly to the PD-1 protein, more tightly than the natural ligand, which is PDL2. There's a lot of interest in this protein for cancer immunotherapy, and we're starting to think in those directions. So coming back to a theme that Jim mentioned first, and then Andreas talked about, what about specificity? And so we thought we would go for would be cases where you have a protein family which has very, very similar members, and to see if we could design proteins which distinguish them between them with very high specificity. And I'll give you 2 examples. The MDM4 and MDM2 proteins, which target P53 for degradation. There's a collaboration with Michael Dyer and Brenda Schulman at St. Jude. And then in the BCL2 family, which there are many, and these have been mentioned already before, involved in apoptosis, we sought to design a protein that bound very specifically to one particular family member. Disrupting these interactions selectively is hard to do with small molecules, and since it's intracellular, it's hard to do with small molecules also. So this is work by James Moody. These are MDM2 and MDM4 superimposed. They're very, very similar proteins, which is why it's hard to distinguish them. James designed this protein shown in blue, which has something that's sort of like the p53 helix, that's what gets recognized, but then has this whole extra part here that cradles the surfaces of MDM2 and MDM4. And with this design, he can distinguish, here he has a binder that's binding MDM4 170-fold more tightly than MDM2. He's also made a version that binds MDM2 much more tightly than MDM4. And the St. Jude groups have shown that these proteins selectively bind the cognate but not the non-cognate target in cells. And so they're using these to probe the function of MDM4. And the preliminary results they're quite excited about is that binding specifically to MDM4 but not MDM2 causes apoptosis. But that again is still early days. On the now, I mentioned the BCL2 family proteins. Here's the BIM peptide, which you've already heard of, bound to BCL2. And Eric Proko sought to design a protein which would bind very specifically to the Epstein-Barr virus protein BHRF1, which is thought to be an anti-apoptotic protein. And so if you look at the BIM peptide, it binds to BHRF1, but it also binds to many, many host proteins. So if you use this peptide to try and, if you add it to cells that are infected by the virus, you get all this crosstalk from binding, this is nanomolar, binding to these self-proteins. However, the protein that Eric's designed binds very much more tightly to BHRF, the viral protein, than it binds to any of the host proteins. I should say that Eric's gone on to use a similar strategy to make specific binders for a couple others, including MCL1. So when Eric adds this protein to, I should say this collaboration with Pat Staten's group at the UW, when he adds this protein to mitochondria from infected cells, he adds it to mitochondria from uninfected cells, there's little effect, but from infected cells, there's considerable cytochrome C release. However, when you add just the BIM peptide, you get a strong response in the uninfected cells because they have all those other host BCL2 proteins in there, and you get less response. It's been known for a long time that these cells are resistant to apoptosis. So if you deliver the protein into cells, you see a similar thing. Here, these are uninfected cells treated with a construct with Eric's protein on it. Nothing happens, but if you add it to infected cells, you get selective decrease in viability, which is not seen with a protein of related size and amino acid composition. So, of course, this is done in cell culture. The general challenge, of course, is delivering this protein to the cells for real use in vivo. All right, now moving on to small molecule binding. This is, again, we can approach this problem in a similar way. This is a model small molecule that Christy Tinberg and Sagar Kar chose as a model for studying this protein, and they designed a protein that had that was shape complementary to the ligand, which is shown here, and interacts with it via very specific designed hydrogen bonding interactions. And Christy made the protein and showed that it bound the ligand, and the crystal structure solved in Barry Stoddard's group confirmed the design model. Here you can see the purple is the design model, and the teal is the crystal structure, and you can see they're very, very similar, and that these designed interactions are in place. Now, what's neat about it is that, again, Christie designed in these specific hydrogen bonding interactions. If she surgically removes these substitutions, these interactions which were designed in, she can reprogram the specificity of this design for other related steroids, and in a very predictable manner. So we've used this general method now to design small proteins that bind to a variety of different ligands that are of interest really for two reasons. One is for diagnostics. I should say that this molecule is used to treat certain types of heart conditions, but has a very narrow therapeutic window. So it's very important to be able to know how much is in somebody and then to soak up the excess if there is too much. And that's something, so we're thinking about these proteins as therapeutic sponges. And so where there's a series of small molecules, such as some of the latest blood thinners, which have this property that too much is really bad. And we're designing binders to these. Okay, so I want to close by discussing the design of protein assemblies. This is work by Neil King and Jacob Bale and Will Scheffler in the group. So they've developed a general method for assembling from proteins that will adopt, I'll just fold up to these highly symmetric architectures. This is a single protein subunit and these are cryo-EM, these are actually negative stain EM micrographs, and these are averages, and they look like little dice. I always want to try and pick them up and roll them. We can't quite put different numbers on the different sides, but we're getting closer, as I'll show you in a moment. These structures, these designs, what's neat about these design structures, which have octahedral symmetry, and the ones I'll tell you about in a moment, which have more complex symmetries, is that they crystallize within hours of setting up trays. It's because of their high symmetry. This shows crystal structures of two of these designs down the different symmetry axes. And again, they're almost identical to the design models. Now, these designs are made from a single type of subunit, but and so what Neil and Will and Jacob have moved on to since more recently is designing cages from two subunits. And here's the basic idea. This is, imagine two inverted tetrahedra and placing a trimer at each vertex of the green tetrahedra and another one at the vertex at each vertex of a purple tetrahedron. And then you have various degrees of freedom. You can spin these tetrahedra around their these trimers around their axes. You can move them in and out. We search this space and identify arrangements where there's a lot of complementary packing between the subunits. And then we use this type of design calculation I showed you in the first movie to optimize the interface between them. These proteins, again, as with everything I'm saying, I should emphasize we make a number of designs, only a subset of them actually fold up as desired, in this case, form the higher order assembly. And these are cases like that. So here are 5 different examples. It's a little bit hard to see, I think, but when you look at a negative stain EM field for these designed assemblies, you see fields of particles which are extremely homogeneous and look like these structures. And Todd Yates's group has been able to solve structures for 5 of them now. And when you compare these structures down the different symmetry axes, they're again very similar to the design model. The design models are here, and here's the structures. And here's another case. So you can see we can design these types of 2-component materials with very high accuracy. Okay, well, what are these good for? Well, there are a number of applications, which I'll just spend a minute or two describing. I talked first about these 2-component cages, which are shown in green. 1-component cage is shown in green. These 2-component cages, of which I've shown you ones like this and this, are much more versatile. I didn't show you here, but these 1-component cages assemble inside cells. But for these 2-component cages, you can make the components separately and then mix them together. You can even mix together extracts, and they assemble in the extracts to form the desired structure. One application we're very excited about is targeted delivery. We can put different types of molecules inside, and we can put targeting domains on the outside. So we can really customize the particles for really any design property. Because these proteins, these assemblies essentially we're designing to spec. So any type of property you'd like to have in a target delivery vehicle, you can design in. I'm not going to show you, but we've used similar techniques to make two-dimensional arrays. Then the final application, which is perhaps the easiest one, is to start thinking about vaccines. And this is a topic which has come up several times. What I'm showing here is a stabilized GP120 trimer developed by John Moore and coworkers over the years. And what we're doing is we have this two-component cage, and we have these cages with different assemblies. Remember, Pamela pointed out that the problem with HIV is that you have, in the natural viruses, you just have one or two spikes. But here we can control very precisely the valency and the spacing by using different of these two-component arrays. And then this is just one component that the GP120 is going on. The other component, we're exploring different possibilities for triggering the innate immune system, so to have sort of a rationalized adjuvant. You could even imagine packaging small molecule adjuvants inside. So we're very excited about this area. And I think I have a couple more minutes left. So I just wanted to show you one fun example, another fun thing that protein folding game players have done. These are real, they've actually done stuff. It's not just make protein art. And so in work I didn't tell you about, we spent a lot of time designing enzymes. And so one enzyme that we designed was for the Diels-Alder reaction, which makes two carbon-carbon bonds between these two substrates. This was kind of a neat reaction to work on because there aren't naturally occurring enzymes known that catalyze a bimolecular Diels-Alder reaction. But like the other enzymes we've designed, it was a pretty lousy enzyme. This is activity as a function of time, and the units are wrong for it. They're not what we would like. So we thought, well, maybe we could give this out as a problem to Foldit players. So here's the designed enzyme, and when you look at it, you can see why it's probably not a very good enzyme. The Km for the smaller of the two substrates is 100 millimolar. It's horrible because there's nothing really interacting with it. So we gave this out. We called this Cover the Ligand as a Foldit challenge. And here's what Foldit players came up with, this crazy long helical hairpin insertion. And Chris Ivan was an undergraduate in the lab who was brave enough to actually test this out. And it actually gave a rather considerable increase in activity for this. It's the best known catalyst of this reaction known to date. And when the structure was solved, it turned out they'd gotten it right for the right reason. This was solved in Barry Stoddard's group. So people in the general public can come up with really off-the-wall solutions that a card-carrying, biologist or biochemist, whatever, might not think should work, but sometimes they're right. And so this is why we're excited to continue to work with this community. All right, so the acknowledgements, of course, the most important part. I've been fortunate to be working with really fabulous people. Novo and Rie Koga did the work on the new globular protein design. Vikram again, who was a student with Avis Chakravarti here, who is doing this fabulous work on small peptide design. Neil and Jacob and Will designed those self-assembling materials. Sorel and Tim and Aaron did the work on the flu stem binders. Eva did the work on the head region binders. And I also wanted to mention, for all the antibody people here, that she's designed a really neat pH-dependent Fc binding protein, which binds with very high affinity at pH 8, but doesn't bind at pH 5. So if you want to purify your antibodies under gentle conditions, get in touch with me or her. Eric did the work on the BCL2 family binding proteins, Cassie on the PD1 binders, and James on the MCM, MDM2, MDM4. And Christy and Sagar did the work on small molecule binding, and we've been fortunate to have wonderful collaborators on the structure side.